Assalamu alaikum, dear guys. This is paper chromatography, and today is Friday, and the lab was expected for Chem 100 student G section. So in the last lab with the B section, we did 3 different labs. One was the sublimation of naphthalene. Another was the separating the mixture by using the process of sublimation. And number 3 was the paper chromatography, how to separate the mixtures of different inks by using paper chromatography. These were the 3 labs and experiments we did on Monday. So now I'm going to explain to you, and the activity related to these 2 labs is I will upload, and you have to follow the submission dates, because after that, once the window is closed, you are not be able to upload your work. Now, paper chromatography. First of all, I will give you this information that this paper chromatography is actually a analytical method, okay, which is used to separate colored chemicals or substances, or we can use, it's a powerful analytical tool. And the main thing is that this analytical tool requires a very small quantities of the things to be analyzed. So what is actually the main purpose of this paper chromatography? The main purpose for this chromatography, that it's a technique for separating dissolved chemical substances by taking advantage of their different rates of migration. If you have the mixture, for example, if you have the mixture, okay, and you don't know what is in the mixture, right? And you don't know what is in the mixture, you just put the drop of the mixture, okay? And this is the filter paper, right? This is the filter paper. You just put a drop of mixture over here, and then put this filter paper in a tank, okay? Tank which contains solvent, okay? और आप clamp कर देते हैं इस paper को in the solvent in this way that the tip of the filter paper needs to be dipped in the solvent. Now this mixture, as the solvent move this filter paper, it will go for a certain distance, mark that distance. Meanwhile, this is also separated. For example, like this, okay? So this is actually the in which we need very small, quantities, right? It is an inexpensive but very powerful analytical tool, and you need only very small quantities to analyze them, okay? So next, we need to understand what is paper chromatography and how does it work. So the paper chromatography, in this type of separating techniques, substances are distributed between a stationary phase and a mobile phase. Now the paper is the stationary phase and the solvent system is the mobile phase. So when a colored chemical sample is placed on a filter paper, the colors separate from the sample by placing one of the end on the paper in the solvent. So that's the way it works. Then we come to the principle. What is the principle of paper chromatography? So principle of paper chromatography is based on one thing which is very important, and that is the liquid partition chromatography. Liquid, just let me write it. Liquid partition, P for partition and C for chromatography. So LLPC, liquid partition chromatography. In paper chromatography, especially the quality of paper is very important. इसको हम कहते हैं वॉटमैन फिल्टर पेपर, और इसका नंबर जो होता है दैट इज नॉर्मली वी यूज दैट इज द 42. So 42 is the number of filter paper, and the name of the filter paper is Whatman. So in paper chromatography, special quality paper known as chromatography paper contains water trapped in it, which acts as the stationary phase. जो पानी होता है बीच में, because the solvent mixture we usually take is a mixture of two or more than two solvents. तो इसमें जो water होता है वो stationary phase में absorb trap हो जाता है. The solvent rises up the paper by the action which is known as capillary action and flows over the spot. अगर आपसे कोई पूछे कि how does this water, this solvent system moves up to this paper, so you can say that it is according to the capillary action. So what are the types of paper chromatography? We will discuss later on. Now we come to this. This is the simple paper chromatography with various inks. For example, if the drop was if in the last slide we put the drop of the mixture of the inks, now when you put this in the solvent, it will appear like this. So this is a very beautiful, picture of paper chromatography. Now this is the outline of the paper chromatography. So आपका task क्या है इसमें, क्योंकि जो 100B के students थे, they did in the laboratory. Okay, so they have the strip of chromatography result. Now what you do, you copy this thing, you take this portion, okay, this is the portion, right? You take this portion and draw something like this. Do not go for the cut and paste. I need to be done with handwriting, okay? So आपने एक पेपर लिया, उसके ऊपर आपने यहाँ पर जहाँ पे ओरिजिनल लिखा है, यहाँ पे आपने dot spot, you need to show the spot of the mixture, and this is the solvent front, right? उसके बाद red, purple and blue. So right, ये आपके drops हैं, और इनका आपने RF value calculate करनी है with handwriting, not by the computer. RF value. RF value is the distance traveled by the separate red dot divided by the distance traveled by the solvent. So ये आ जाएगा, for example, solvent जो है आपका, ये हम ले लेते हैं 10. ये this is 10 centimeters, okay, this is 10 centimeters. So if we need to calculate the RF value of this red, so it traveled only 2 centimeters, so 2 divided by 10, this will give you is equal to 0.5. You got it? So this is the RF values. In this way, you calculate the RF values of all these three blue, purple, red ink, and then you take the picture and then you upload that picture on the Moodle. So उसमें मैं तमाम options कर दूंगी कि आपकी pictures जो हैं वो भी accept हो जाएं. अब question ये है कि ज़्यादातर where we use this paper chromatography. So it has become standard practice for the separation of complex mixtures of amino acids, peptides, carbohydrates, steroids, purines, and a long list of simple organic compounds. Inorganic ions can also readily be, we can separate on the filter paper. Principle, what is the principle? The principle involved, I have already explained you कि part इसमें जो है क्या कहते हैं, जो प्रिंसिपल है दैट इज एक्चुअली कैपिलरी एक्शन है सारा, ठीक है. इसमें 2 phases हैं आपके, वन एक मोबाइल फेज है, वन एक स्टेशनरी फेज है. फिल्टर पेपर इज द स्टेशनरी फेज, सॉल्वेंट सिस्टम इज द मोबाइल फेज. How does it work? Substances are distributed between the stationary phase and the mobile phase. So when a colored chemical sample is placed on a filter paper, the colors separate from the sample by placing one end of the paper in a solvent. So this is like this. It works. Then principle involved is the partition chromatography, where in the substance are distributed between the partition between liquid phases and लिक्विड फेस और उसकी जो पेपर की फेस है, ठीक है, और ये इसका प्रिंसिपल है. नेक्स्ट, what are the types of paper chromatography? Two types की है, ascending paper chromatography and two dimensional paper chromatography. जो ascending अभी जो what I have explained to you, that is the ascending paper chromatography. What about the two dimensional paper chromatography? First of all, you go for the ascending paper, then take out the paper, dry it, and then turned turned through 90-degree, and then again put it in the solvent tank. ठीक है, और ये टू टाइप से, टू एंगल से आप इसको करते हैं. The two main types of chromatography are gas liquid chromatography and gas solid chromatography. So again, this is the diagram, this is the homework, this is the work, this is the target, this is the activity of 25%. What are the applications of chromatography? To study the process of fermentation and ripening, to check the purity of pharmaceuticals, to inspect cosmetics, to detect the contaminants in drinks and foods, to examine the reaction mixtures in the biochemical laboratories, and to determine dupes and drugs in humans and animals. So these are the main factors, right? So RF value kya hai? The distance traveled by the separating spot divided by the solvent front. So you will get the RF means retention factor. RF value is the distance traveled by a given component divided by the distance traveled by the solvent front for a given system at a known temperature. It is a characteristic of the component and can be used to identify components. So RF value are very important. So this is all about the chromatography. So I hope I will explain you the physical and sublimation process also, but today is Friday, so I will do that work after namaz. Okay, so thank you so much.